Very good evening to one and all. Welcome to Pharma National Webinar Series on Role of Cytokines in Immune Inflammatory Diseases, organized by Vignan Pharma Institutions. Myself, Dr. Purna Nagasree, working as an Associate Professor from Department of Pharmaceutical Chemistry, VIPT Visakhapatnam. First of all, I would like to thank Dr. Aparanji for accepting our invitation. Thank you, ma'am. Now it's my privilege to welcome the Chief Patrons, Dr. L. RATHAIAH Garu, Chairman of Vignan Group of Institutions, and Sri L. KRISHNADEVARAYULU, Vice Chairman and Honourable Member of Parliament for Loksabha. I would like to welcome the patrons, Dr. K. RAMAMURTHY NAIDU, Chancellor VFSTRU, Dr. M.Y.S. PRASAD, Vice Chancellor VFSTRU. I would like to welcome the Co-Patron, Dr. M.S. RAGHUNATHAN, Registrar VFSTRU. I would like to welcome the heads of the institutions, the organizing heads behind this webinar, Dr. Y. Srinivasa Rao Garu, Vignan Institute of Pharmaceutical Technology, Visakhapatnam, Dr. P. Srinivasa Babu Garu, Vignan Pharmacy College, Badlamodi, Dr. M. Srinivasa Murthy Garu, Vignan Institute of Pharmaceutical Sciences, Hyderabad, and Dr. C.H. JITHENDRA Garu, VFSTRU, Guntur. I welcome all the faculty members and student delegates to grace the webinar event. Welcome one and all. Now I would like to invite Dr. Y. Srinivasa Rao Garu, Professor and Principal, Vignan Institute of Pharmaceutical Technology, Visakhapatnam, to give his introductory remarks. Dr. Y. Srinivasa Rao Garu.
Thank you, madam. It is my immense pleasure to take this opportunity on the occasion of conduction of webinar on role of cytokines in immune inflammatory diseases given by the Dr. P. Aparanji Madam Garu, Associate Professor, Andhra University, Department of Biochemistry, and my beloved head of the institutions, faculty members, and my dear students. Very good evening to all of you. Today, we are going to giving one of the prestigious webinar, that is cytokines in human inflammatory diseases. So during the pandemic situation, so many during that period, that means April 23rd to May 15th, so many deaths occur in India across for a day around 3,000 to 4,000 people. So during this period, these cytokines, one of the important major concern that play an important role. If we have to see the situation for first wave and second wave, lot of differences is there, mainly due to the because of role of cytokines. And these cytokines are a crucial component of human response, as they aid in the intracellular communication and are involved in both health and disease through their physiological and pathological effects. So they are glycosylated small polypeptides synthesized and released by a variety of sources, including lymphocytes, leukocytes, and other cell sources. They are involved in mediating natural immunity and regulating the activation, growth, and differentiation of lymphocytes, leukocytes, as well as other cell types. So these cytokines play a role in the regulation of the immune-mediated inflammation and stimulate hematopoiesis. So their effects are mediated via cell surface receptors based on their physiological and pathological effect. They are ideal therapeutic agents either by themselves or as anti agonists, which also include specified monoclonal antibodies directed against them. This type of importance is there today. The same web seminar of cytokines in the immune inflammatory diseases. Once again, I thank to the madam to accepting our invitation, given the today webinar topic on cytokines in the human inflammatory diseases. Thank you for giving this opportunity. Thank you. Hello.
Introduction. Now we have invited Dr. S. Lakshmi, Professor of Department of Biotechnology, Pharmaceutical Technology, to introduce to Dr. S. Lakshmi.
Thank you, ma'am. It is my great pleasure to introduce today's speaker, Dr. Aparanji Posarla. Currently, she is working as an assistant professor in Department of Biochemistry, Andhra University, Visakhapatnam. She has more than 15 years of teaching and research experience. Her research areas are immunology and molecular biology. She is expert in study of autoimmunity disorders and cytokines. She has 2 patents. She received 2 research grants from DST and DST Serve Fast Track. Handled 3 consultancy projects from Girijan Cooperative Corporation, Andhra Pradesh government. Her achievements are achieved BSc Gold Medal from Mrs. Savian College, Visakhapatnam in 1993. Qualified in UGC GATE 1995 and obtained Junior Research Fellow and Senior Research Fellowship. Received Young Scientist Presentation Award at AIMS in 2005. Received DST Women Scientist Award in 2009. Received Travel Award from Indian Immunology Society for presenting research paper in Asian Congress on Autoimmunity held at Singapore in 2009. Received Gold Medal for Senior Scientist Oration in 2010. Received FIMSA 2012 International Award at New Delhi. Received Fast Track Young Scientist Award from DST in 2013. Awarded Senior Research Associateship from CSIR in 2013. Received Associate Fellow Award from AP Academy of Sciences, Amravati in 2017. Recipient of Travel Bursary from CSIR and Immunology Foundation to attend for FOSIS 2018 annual meeting held at San Francisco, USA. Awarded first prize in Novel IP Insights Innovative Challenge 2K18 for anti-arthritic compound and its making process in 2019. Awarded free patenting for exemplary submission of 2 research ideas by Novel IPR Academy in 2019. She has MOU with Kalam Institute of Health Technology and Andhra Pradesh MedTech Zone Visakhapatnam for intellectual property technology transfer of portable biosensor for type 2 diabetes in 2020. In addition to these achievements, she also served as executive member for Indian Immunology Society, Department of Biochemistry, AIMS, New Delhi from 2015 to 2017 and 2017 to 2019. Organized World Immunology Day 2015 and 2016 under Indian Immunology Society Cmde and National Seminar Department of Biochemistry, Andhra University. Received travel grants from various funding agencies like DST, CSIR, and Immunology Foundation. She was invited as a speaker for conferences organized by various institutions like Asian Congress on Autoimmunity at Singapore, NIT Warangal, NIMHANS Bangalore, ICMR Institute Patna, Geeta Medical College Visakhapatnam, and Nirma University Ahmedabad. She is serving as editor and reviewer for various national and international journals. With these brief introductory remarks, now I welcome today's speaker, Dr. Aparanji Posarla. Welcome, ma'am. Over to you, ma'am.
Now I invite Dr. Aparanji ma'am to take over the session. Thank you.
Thank you, Dr. Satyalakshmi Garu, for your nice introduction and words. And I would like to thank SRINIVASA Garu SRINIVASA garu, and especially Dr. Purna Nagasree madam, and Vignan College of Institutions and Organizing Committee for providing me this opportunity to share some of my research experiences and my contributions. I would always be indebted to Andhra University for providing me a wonderful platform to carry out my research and also contribution towards research. Today's, shall I share my presentation? Purna, shall I share my presentation now?
Yes, ma'am.
Okay. Yes. Today's my topic is role of cytokines in immune inflammatory diseases. As you are aware that inflammatory diseases, inflammation, actually it is a good phenomenon for any diseases. In order to protect our body, the first symptom comes this inflammation. So what are inflammatory diseases? Inflammation has been suggested to be the principal cause of chronic illnesses such as diabetes, obesity, rheumatoid arthritis, neurological disorders. So always inflammation is associated with any of the diseases. If you consider any disease, inflammation is common. Actually, for a specific cause, inflammation is a defending mechanism. So where you will see the cellular infiltration at the site of inflammation, whenever a specific cut or injury happens, immediately the immune cells, other cells, comes at the site of infection or injury and defending against the microbe or and protects the body against the microbes. Microbes could be bacteria, fungi, or virus, whatever. And this inflammation, not only for infectious diseases, this inflammation is also could be due to other diseases like autoimmune diseases or inflammatory and said to be immunoinflammatory diseases. The best examples I can give: rheumatoid arthritis, psoriasis, and multiple myeloma, multiple sclerosis, and other diseases where the inflammatory bowel disease, the inflammation is the common cause. So my research mostly involving the cytokines. I'm fascinated about these molecules. Today, you know, the buzzword is inflammatory cytokines. There is no specific treatment for all the cytokines which are produced in the cytokine storm in COVID situation. The alarming situation we faced during second wave. And there is no single specific medicine which treats all the cytokine storm.
Kindly excuse ma'am, Aparanji ma'am. Your screen is not visible, ma'am. Slides are not sharing.
Slides are not changed. I did not change the slide. The same slide. Okay.
Just share the screen, ma'am. Our screen is not visible, ma'am. It is not shared, ma'am.
Okay, sorry. 1 minute. Is it visible?
Yes, ma'am. It is, ma'am.
Okay, now, is it okay, Purna?
Yes, ma'am. It is, ma'am.
Okay. Yes. So, the immunoinflammatory diseases. So, I have analyzed and estimated, especially we have analyzed and estimated cytokines like mostly inflammatory cytokines, IL-1 beta, IL-6, interferon gamma, IL-8, IL-33, interleukin 12, interleukin 9, interleukin 2, TGF beta, which is a very important cytokine. IL-17 is a pro-inflammatory cytokine, which is a recent cytokine. And another cytokine which we have estimated in psoriasis is IL-9. That is the recent cytokine. IL-23, IL-4, TNF, alpha, IL-10, and IL-17. These are the cytokines we have studied and estimated during different conditions, disease conditions, immune inflammatory conditions. What is the reason for this immune inflammatory conditions? Actual reason is the autoantibodies actually recognized in case of autoimmune diseases. The autoantibodies produced, produced in the body, which recognizes the molecules, patterns, like disease-associated molecular pattern molecules, which are mediators, and these dams activate myeloid immune cells with an enhanced inflammatory immune response. So leading to abnormality as an exacerbation that leads to damage of the host tissues. Actually, this is the main pathogenesis which is involved in any of the immune inflammatory diseases. So when you consider cytokines, you all know that as said by SRINIVASA, and these are simple polypeptides or proteins which are involved in cell-cell communications, which are produced in response to, which are produced by the cells, immune cells, and some other non-immune cells in response to microbes and other antigens that mediate, these cytokines mediate, most importantly, cell-cell communication, and they also regulate a cellular network and regulate immune and inflammatory reactions. These cytokines, as you know very well, links innate and adaptive immunity. And in order to exhibit their functions, they require receptors. That's why they're considered as local hormones. Regarding these cytokines, the difficulty in working cytokine area is because these cytokines are produced by one kind of cells and exhibit their action on different cells. And the same cytokine will be secreted by the other cells, the different kinds of cells, and exhibit different activities. So they exhibit pleiotropy and redundancy, as you know very well. And this is the difficulty with cytokines. When you understand cytokine network, it is the mechanism of action would be very, very difficult. One cytokine regulates the action of the other cytokine, so we need to understand the detailed understanding of mechanism of action of these cytokines in particularly immune inflammatory diseases. So what is the exact cause for production of these immune inflammatory diseases? Excessive production of actions of cytokines can lead to pathological consequences. The administration of cytokines or their inhibition is a potential approach for modifying biological response associated with the immune inflammatory diseases. So potential research, that's why we are in search of natural products can be used for the treatment or a modulation of these immune responses. We have tried in our lab both natural products, marine and plant origin. And this is very good slide. From this, you can understand. So homeostasis always maintained for the immune system, especially if it exceeds the immune system is overexpresses. What happens? Suppose if it is for internal threat, then it leads to autoimmune diseases like Hashimoto's thyroiditis, rheumatoid arthritis, systemic lupus, inflammatory bowel disease, type 1 diabetes. It comes under where you can see, what are autoimmune diseases. So here I worked on rheumatoid arthritis, animal models, and come to if the hypersensitivity. So immune system is overexpressed. The hypersensitivity responses will be getting. You can see in allergic reactions, food sensitivities, allergies, eczema, and asthma. So when immune system is under reactions leading to the cancer like hepatitis, HIV, shingles, tuberculosis, where you can see the intracellular pathogen is intracellular in these aspects. Okay, the TB, when you consider tuberculosis, the main problem for the treatment of tuberculosis is the pathogen remains within the macrophages, intracellular. The treatment would be very, for such diseases, the treatment would be very difficult. The inflammation common for all these diseases, and if it under reaction, the infection, the other type of reactions are and diseases are infections caused by bacteria like mold, fungus, parasite, and viruses. Okay, so we would like to focus mainly on cytokines here, and so what I mean to say from this slide, the balance homeostasis of immune response has to be maintained, normal healthy individual. Whenever it inclined for any of the reason, the abnormality arises. Modulation of immune response. As you know very well, immune response is a protective immunity. So when you consider protective immunity against microbes is mediated by the early reactions of innate and later responses are called adaptive immunity. Two types of innate and innate means by birth. The immune system is coming from by birth, so that whenever the microbe enters, immediately it eliminates, which is nothing but a non-specific immune response. And adaptive means a very specific immune response, and mainly the cells which are involved in the adaptive immunity is T lymphocytes. The differences, the main differences between the adaptive and innate, the stimulus for innate immune response is lipopolysaccharide. In that way, you can differentiate the endotoxin present in the lipid portion of the lipopolysaccharide and the peptidoglycans. These are the antigen stimuli for innate immune response. Mostly T-cell derived, you will get T-cell derived cytokines are produced, and this happens in innate immunity. When you consider adaptive immunity, only the stimulus are nothing but exclusively proteinous antigens. So you know what is here in this slide, the modulation of immune responses. We wanted to say with the help of natural products, we use medicinal plants and marine natural products for modulating the immune response in innate and adaptive means. And mostly innate is innate during the development of immune response, what happens, you know what is primary response and secondary immune response. And secondary immune response, you will have a memory response, that is the trained immunity. Innate immunity has to be first elevated whenever the pathogen enters, and then subsides. And immediately, whenever immediately after declining the innate response, the adaptive immunity has to be raised. Then only it controls the immune response with the help of adaptive immunity, because adaptive immunity is the most specific one. Innate immunity is the non-specific one. That's how it controls and regulates any of the disease. But what is happening in COVID disease? But innate only it is due to the damage. The entire damage is only due to this innate immunity, innate immune response. The innate immune responsible cytokines are nothing but IL-1 cytokine, TNF, and IL-12, and interferon gamma. These are the cytokines mostly involved in innate immune responses. Adaptive cytokines are IL-4, IL-5, IL-2, interferon gamma is also there, the adaptive immune response. But what happens in this, during COVID, the innate is continuously elevating. It is not declining it at all. After declining, immediately the adaptive response has to be raised, more specifically in order to eliminate the pathogens. Okay, but it is not happening. So during this period, the pro-inflammatory cytokines and other inflammatory cytokines like IL-6 and IL-6, IL-1, these cytokines will be produced in excess, leads to the cytokine storm. So with you can modulate the immune response in such disease, inflammatory diseases, either through epigenetic modifications and metabolic reprogramming and long-term effects and other altered T-cell responses, altered responsiveness. So especially we followed altered T-cell responses we observed altered T-cell responses by administering this natural products, either in the form of plant extracts or in the form of marine extracts. So a research focus mainly on first, in order to carry out any research or a research project, first you need to have, particularly in the field of immunology, you should have a thorough understanding of the disease and how the disease progress will be, and what is the role of immune system. And the immune system means comprises immune tissues, cells, organs, and immune molecules. So you know, T-cell molecules are nothing but cytokines, and B cells are antibodies. You have to look both the humoral and both arms of immune response, humoral response as well as the cell-mediated immune response, and other mostly in order to understand the disease progression for any of the disease. And the second point, you should concentrate on genetic expression, analysis of the genetic expression. These are also very important because, as I said, all the immune molecules are under the control of genetic constitution. Your individual, the genetic constitution of the individual, as you know, it varies from one to another person. So what happens? The MHE will present in the organism will decide whether it has to associate with particular molecule or not. So genetic expression of the molecules are also very important, how it reacts. So you need to understand the genetic mechanisms and genetic expressions of the molecules. And also you need to understand signaling molecules, signaling pathways, and what are the role of infectious agents. Normally, for any disease, in research laboratory, we will study, will induce a disease, we'll have a disease model in our hand, we'll particularly select some parameters, and we'll screen the compounds against those models. Actually, you should have a prior art. So what is the, in your hand, so what is the background of that particular disease? Okay. Is there if I'm working with rheumatoid, this is my experience, that's why I'm telling. And when you consider rheumatoid arthritis, so I used animal model for rheumatoid arthritis. So what happens that for that, if there is any background, infectious background for the development of the autoimmune disease, any pathogenetic role, any etiological agent which is responsible for the pathogenesis of for the development of disease or induction of the disease. You should have a prior idea about this causative agents which produces diseases in the animal models or any other diseases. Okay, and this is the role of any other infectious agent for the development of immune inflammatory diseases. Next, studies on animal models. Animal models, particular standardized animal models should be there for the induction of the diseases. And you have to select a suitable animal model for the development, for the screening of your compounds. And the effect of therapeutic intervention and prevention, you have to talk them. These are the important points has to be considered for any scientific research. And coming to actually immune inflammatory, what are the immune causes, root causes for immune inflammatory diseases? We have to find out. In order to treat patient, we have to find out first what are the root causes for any disease. So the causes for the immune inflammatory disease involves first genetic cause, that could be a major histocompatibility complex. You know what is the role of major histocompatibility complex? That the role of major histocompatibility complex is here is which is mostly expressed by the, as you know, by the antigen presenting cells. There are two types of MHC, class 1 and class 2. Class 2 molecules, which are the nature of MHC class 2 proteins, they're protein in nature, they're expressed on the antigen presenting cells, which could be a macrophages or dendritic cells or any other epithelial cells, and sometimes B cells expresses these MHC molecules. If the antigen presenting cell, here the antigen presentation is also very, very important. Most of the people will not screen against your drugs, not screen against this antigen processing and presentation. That is very important one. So how the drug is taking, how the antigen is taken by the host, that is very important in order to screen your drug. So here MHC plays a very, very important role. How during the presentation, what happens? The MHC presents its antigenic peptide after processing, present its processed peptide towards T helper cells. If it is MHC class 2, if it is MHC class 1, it presents its peptide antigen towards the CD4 cells for cytotoxicity. What T helper cells? Finally, it forms a trimolecular complex in order to get the cell-mediated immunity, as you all know. So, if T helper cells get activated, what will happen? And signals goes to B cell and produces antibodies. If T cell, the cytotoxic T cell, get activated by the formation of this trimolecular peptide, and the cytotoxicity of the cells takes place. That's how it protects the host in any of the diseases. So you need to study the antigen processing and presentation studies, which gives you a lot of information and how the this process is taking place and during the disease progress. And the next point, important, environment. There is a role for environment. It could be a sunshine, stress, chemicals, drugs, etc. For the which also involves and causes also causative factors for inflammatory diseases. And next, immune regulation. And immune regulation is through cells, cytokines. The third thing is that these are the important regulators and mediators of inflammation and diseases. So we should understand in detail about the cytokine secretion and the cytokine action, mechanism of action, how they are acting as a regulatory molecules. Then you will understand the entire mechanism, detailed mechanism of the pathogenesis of for a particular disease. And viral trigger. So you need, in order to produce or secrete cytokine by the cells, immune cells. So external trigger must be there. That could be a virus, a microbe, or any other fungal, or any other sometimes any other super antigens, microbial antigens acting as a super antigen. That's why cytokine storm and the septic shock. These two are the reasons for the COVID. They are telling that these two are the reasons for the second wave majorly affecting the people. So the septic shock is mainly because of the super antigens, which triggers the immune cells, especially T cells. unknowingly, we don't know the exact mechanism for this stimulation. The T cells get stimulated. What happens immediately? They are unknowingly proliferate, produce more inflammatory cytokines and also pyretic cytokines like IL-1, inflammatory cytokines like TNF-alpha, IL-1 beta, etc. So these will be produced continuously. When these will be continuous, what will happen? And the same condition what we are facing with the COVID. So continuously the cytokine storm will be there in the system. That is due to septic shock. Super antigens will be, some bacteria produces these super antigens, especially endotoxins. Endotoxins will be acting as a super antigens. And these are the viral external trigger also stimulating the immune network to produce and secrete cytokines. These three together, in an amalgamation of all these three factors, are the responsible for the development of immune inflammatory diseases. Okay? If you consider, while studying the mechanism, if you consider these three, then you can do anything. You can do wonders. So actually, we screened our natural products from plant origin or marine natural products on diseases on animal models like rheumatoid arthritis and human psoriasis and also human malaria and animal model for malaria. These three diseases I have worked on. So when Purna asked about my research slides, actually I would like to present on a different topic, COVID, and she asked me to share some of my research slides so far what I have done. So they immediately I changed within 2 days and prepared my slides. I compiled all the data and for my presentation. Okay. So first let us start with the rheumatoid arthritis, the first disease which I have worked in during 1996. Okay. When I joined the Ph.D. in Andhra University, Department of Biochemistry, under the guidance of Professor Rathod. And this is a wonderful work, rheumatoid arthritis. As you know, rheumatoid arthritis is an autoimmune disease. This is the outline of my research slides. First, we'll discuss about the rheumatoid arthritis. And we have taken two models now. Collagen-induced arthritis, so induced in DBM mice. Actually, these DBM mice we procured from National Institute of Immunology, New Delhi. And then we bred it ourselves. Then we have, in our lab, we have ethical clearance and all that. And so that we bred these mice, DBM mice, and we have conducted all these experiments with the CIM model, which is a very good model for rheumatoid arthritis, collagen-induced arthritic model. And we have also done with adjuvant-induced arthritis in Lewis-strain rats. We have screened herbal formulation, which is collected from Paderu. This is a wonderful formulation. Actually, they have been using for treatment of local osteoarthritis and rheumatoid arthritis at Paderu. So that there is a spelling mistake for Plumbago zelanica. Okay, sorry. And this Plumbago zelanica actually is one of the constant present in the root, as a root, one of the constant present in this herbal formulation. And we have purified that using a protocol, standard protocol, and extracted and purified. That is plumbazine, naphthopino. And that has been used for the assay, T cell proliferation assay, screened against T cell proliferation assay and phenotypic analysis by FACS, flow cytometric analysis, and FACS. This is a cell sorting technique for the separation of T cells from the B cells. How many T cells and how many B cells, CD cells, CD8 cells, and B cells, depending on the markers we applied. And we have done that experiment during 1999. And we have estimated cytokines using ELISA R&D kit. So these are the things we have carried out with the rheumatoid arthritis animal model. And then we have done with malaria disease, and we used the animal model, Plasmodium verae, using C57 black strain mice, and we have induced the malaria. This is the animal model for this disease, and we also work with the human malaria, and this is a marker for identification of the disease, PLDH. In Andhra Pradesh population, we have measured this PLDH. This is in vitro method, and next we have carried out spleen T cell proliferation assay. We have isolated the spleen from the animal and dissected the animal spleen, dissected the spleen from the mouse, and we have carried out T lymphocyte proliferation. We isolated T cells and proliferation assay. This mutated thymidine, and then T cells. Why do we require T cell isolation? Because for the production of the cytokines, culture supernatant. After culturing the T cells using the specific media and all that, we then purified the cytokines and estimated. And for malaria and psoriasis, we have used the advanced technology, that is flow cytometric estimation of very specific method. Flow cytometric estimation of the cytokines, that is cytometric bead assay method. Cytometric bead assay method, and because the concentration, as you know, the half-life for cytokines is very less, and especially in the blood, the sera samples, cytokines half-life is very less, and the concentrations are very low. And and for tissue supernatant, they will get the culture supernatants have high concentrations of cytokines when compared to the sera concentration. That's why we prefer culturing the cells and obtaining the culture supernatant for the estimation of the cytokines. Next, psoriasis also we have done, and I'll show the slides. But this actually, I'm thankful to ESL Medical College and for sponsoring me for chemicals and all that culture chemicals for required for the required to carry out all this work in a biochemistry lab, Andhra University. And also I'm thankful to Professor RAGHUNATHAN, who is the famous dermatologist in Visakhapatnam, who provided and who collaborated with the project, and by providing samples and guidance for that work. And then especially for this work, the human skin and blood samples we use in vitro. We have carried out in vitro T cell culture proliferations, and in addition, we have done. We have isolated keratinocytes and culture keratinocytes cells, which are isolated from the skin biopsies. And after culturing, we have estimated, especially the recent cytokines, Th17 cytokines. Actually, he had asked me why can't we establish this technique for estimating the Th17 cytokines, which are very, very important cytokines. As you know, if you see the literature, you will understand what is the role of Th17 cytokines nowadays in inflammation. And it's the same flow cytometry method we used, the cytometric bead assay on BD company. They also, BD people also supported to carry out this to supply these chemicals. And next, in addition, because the genetic association, you always keep in touch with the genetic association with the diseases. We are also interested about CARD14 gene associations with TH7 for the proliferation, which are very much required for the TH17 cytokine secretion. Okay, so these we wanted to identify these gene patterns, which were done by PCR. Okay, for that we have designed primers and patterns. This is the outline of my work, starting from rheumatoid arthritis. As you know, rheumatoid arthritis is characterized by the chronic inflammation of the synovial membrane of joints, resulting in cell-cell interactions and produce and induce pro-inflammatory cytokine production, which in turn activates the release of proteases, especially matrix metalloproteases. Matrix metalloproteases, thus leading to the bone and the cartilage damage and destruction. So as you know, the same because I have worked with this RA, that's why it is exactly similar to the immune response. When you consider the immune response developed during the progress of rheumatoid arthritis, is exactly similar to the same environment, cytokine environment is there in the COVID situation also, COVID patients also, same thing. They are discussing. So it is a kind of because immune-related inflammation. So for this disease, we have for this disease we have used a very good standardized animal model, that is collagen-induced arthritic model. So this you can see the next slide. This slide, in this slide, you can see this is the plumbago zelanica. Actually, we have collected and which is available everywhere. So we have pharmacy scholars have how they isolate and purify the compound for the isolation. But we followed biosolidated fractionation for the proliferation assay and cytokine estimation. There is we identified that with the plumbazin, there is a significant modulation of immune response, especially T cell proliferation and cytokine responses in human mice. So we have carried out different experiments like histopathological studies, radiological studies, and FACS data and T cell proliferation, cytokines. Apart from that, we also measured the anti-collagen antibodies, that is humoral immune response. So, as you know, plumbago zelanica has been used as a medicine to increase longevity, herbal medicine in order to increase longevity and vitality. It is the most important medicinal plant extensively used in herbal formulations. It is chemically rich with diverse content of active compounds such as plumbasin and chitranone, gelanone, and many other useful naphthoquinone constituents as a multi-purpose medicinal agents were present pertaining to plumbago zelanica, and its botanical constants are acting as immunostimulatory, hepatoprotective, hypolipidemic, and hypolipidemic agents. And plumbago zelanica can exhibit varying degrees of therapeutic values in the treatment of fungal, malaria, and bacterial infections, including cancer. There are so many biological activities. That is the importance of this plant. And coming to animal models which I chose for rheumatoid arthritis. So as you know, AAM model and CI model for screening herbal formulations. So actually these animal models contribute valuable insights to the mechanism of understanding of any immune mediated diseases in human, and for the potential treatments and diagnosis also, we need to have a valuable, a very good model for an animal model in your hand. So we have analyzed collagen-induced arthritic model. We have analyzed phenotypically T cells and B cells using markers like CD4, CD8 for T cells, because CD4 is marker for T helper cells. CD8 is the marker for cytotoxic T cells, and CD19 is the marker for T cells. With these markers, we could able to separate the cells, and we have counted the cells using PACS technique. And then TH1, then we have carried out the T cell proliferation assay. Then we estimated culture. After culture, we estimated the cytokines, especially the cytokines like iron having a very important role for proliferation of the T cells, and then interferon gamma. And we also measured the TH2 response, IL-4 and IL-6, IgG2A concentration. We have carried out with anti-collagen antibodies, and also we have seen the different subclasses of antibodies with respect to this plumbasin compound. then actually in case of rheumatoid arthritis, what we observed in a control, there is a elevated response of IgG2A. So when after treatment with this flambazine, the IgG2 levels were come down. That's why we have mentioned here. So the things I have carried out this thing during my PhD work, and I published a paper in this BMC journal, Complementary and Alternative Medicine. It is modulation of T cell proliferation and cytokine responses by flambazine extracted from Nagasree and And coming to the year 90, I think in the year 2009, and GST sanctioned my women scientist project on biomedical and anti-athletic potential of Andaman and Nicobar. As you are aware that the microorganisms are marine organisms are very, very important. Okay, so ocean and this project we have selected, we have screened different marine organisms and extracts collected from the marine organisms. Again, is this the same model, collagen-induced arthritic model, but instead of M.Y.S, so we tried with the C57 black mice. That is the change from my PhD work to the this project work. So why? Because the procuring a M.Y.S is very difficult task. That's why we procured C57 black mice, because the most thing when you understand the haplotype, that is more susceptible for the development of disease. That's why we have chosen that C57 black mice and procured from NIN and Hyderabad. As you know, oceans are the source of immunotherapy, reservoirs of immunotherapy. You can, I can say, the marine flora is taxonomically diverse, biologically active, and chemically unique. So that's why it is an important and an excellent resource. The marine is an important and excellent resource, which offers great opportunities of new biopharmaceuticals. That's why we focused on marine originated bioactive molecules with immunomodulating potential, with practical focus on molecular mechanisms, specific agents with respect to their targets. So these marine organisms, first we have screened against anti-collagen antibodies levels. Black for anti-arthritic activity, and nearly we have collected 40 marine samples and from Andaman and Nicobar, DST sanctioned deposit. And coming to the this modulatory effects, please, there is a noise from your side. Can you please
Yes, ma'am. We will try to unmute them.
There is some disturbance from.
Yeah, participants, please unmute. Please mute yourselves, all of you. We are just muting them.
Okay. Then thank you. Then modulatory, we have seen the modulatory effects of marine sponge products on inflammatory Th17 cells and immunosuppressive T regulatory cells. These two important, so the balance between the Th17 cells and T regulatory cells are very, very important in order to bring about the homeostasis of immune response, homeostasis and immune response. So that's why we have concentrated in Th17 cells, cytokines, and T regulatory cells. Okay. So sensing collagen and arthritis model. So what we have done, marine sponge, especially Stylisha cautery, that is collected from Andaman and Nicobar Islands and was subjected for, say, ISA guided fractionation. So modulation of inflammatory cytokines by marine sponge products represents a possible approach to the pharmaceutical prevention and treatment of rheumatoid arthritis. So new treatments could use the effects of cells on the function of regulatory T cells. So that's why we estimated, especially these cytokines, using ELISA, that is E-biosense ELISA kit, and IL-17, TNF-alpha, IL-1, and IL-6. These are very important inflammatory cytokines. IL-6, TNF-alpha, IL-17, and IL-1 beta. So these not only promote inflammation but also inhibit the regulatory functions. So IL-17 appears as a novel target in T-cell mediated inflammatory response. That's why it plays a very important role in upstream in the pathway of disease process. Use of IL-17 inhibitors could be a way to control the inflammation, but also to restore regulatory T cells. Actually, this work was done in the year during the year 2012. After that, there are I think monoclonal antibodies for against IL-17 also in the market. And this is the in silico. Is the screen is clear? Can you see the slide?
Yes, ma'am.
And this in silico experiments we have carried out with IL-17 and then biopsy guided fractionation for stylisha cautery. And the extracts were actually subjected to column chromatography for a separation of pure compounds by gradient deletion method. The crude extract was chromatographed over a column of silica gel using eluents of increasing polarity of solvents mixers like hexane, ethyl acetate, pure ethyl acetate, ethanol, and chloroform. So we have carried out biopsy guided fractionation, and then animals we used for the development of the for stealing this extracted compounds, and for stealing these extracts we used male C57 black instead of DBMIs, because the same haplotype H2 haplotype, 8 weeks old, weighing around 30 grams were procured from National NIM Hyderabad, and induction and evaluation of collagen induced arthritis in C57 black mice. The mice were immunized with 100 microliters of collagen type 2, that's complete fluid adjuvant by intradermal injection at the base of the tail. And on day 20, after primary immunization, mice were intradermally given a booster dose, and the booster dose of collagen, and along with the booster, the second dose collagen with the IFA instead of CFA. Incomplete fluid adjuvant will be given at the same time. Was given at the same site. The sera was collected 3-week, 3rd week, and 5th week, and 7th week intervals after primary immunization for anti-collagen antibody by using convex ELISA kit. We used a convex ELISA kit, the estimation of anti-collagen antibodies from the sera collected from mouse and estimated the antibodies. Next assay we have carried out is T cell proliferation. So for this we require tritiated thymidine. Actually, this experiment was done with the permission of Dr. D.N. Rao at All Institute of Medical Sciences in Delhi. So we don't have the we don't have the permission to work with the tritiated thymidine. So I have carried out all these experiments at AIMS to Delhi. Effect of stylisha cautery compounds on cornea-induced, the mitogen-induced T cell, splenic T cell proliferation, and collagen-induced arthritic mice using tritiated thymidine was carried out. And then we are also carried out with the histopathological studies and the cytokine estimation. Same pro-inflammatory cytokines, IL-17, IL-1, and TNF-alpha and 6 were estimated using ELISA E-biosense kit. So the results were expressed using statistical analysis, and actually this was the slide which shows. So this is a C50, so inflammation after induction of the disease we got, and you can see this immunization I have explained. The immunization protocol, and then this is the actually stylisha cautery from which we isolated and purified the compounds and histopathological slides. And these are the cytokine graphs. And before that, what we have done in this project with the help of a molecular docking software, we could able to screen some of the in silico screening of compounds, marine already, which are there in the sponges, which we want to like to work. So we have screened some of the compounds using this docking technique, and able to identify some prominent compounds, and then we selected accordingly. We have selected the organisms and protocols. We have done the experiment, and under this research focus on efficacy of marine drugs on the regulation of cytokine network, the pathogenesis of collagen and induced arthritic model. Effect of marine drug purified from Stylisha cartridge was also studied using the T regulatory cells and Th1 responses. So for all these work towards the specific mechanism based treatment for rheumatoid arthritis. And what are the effects of marine drugs on regulation of cytokine network in the pathogenesis of rheumatoid arthritis? So we have seen particularly these cytokines, and cytokine expression also we have studied using this model, and at mRNA level expression of these cytokines we have isolated the mRNA, and the expression was done using the RT-PCR. So imbalance in cytokine network with an excess production of pro-inflammatory cytokines. Using this knowledge, new approaches to RA from marine source are being aimed to correct this imbalance. The next disease is psoriasis. This is also immune inflammatory disease, as you know. There is for this till now there is no specific cure for this RA and for such diseases like RA, psoriasis, etc. So because because of the problem with the abnormal immune response. So first, if we identify the reason for that, then it is very easy to work with the disease. A comparative study was done with lesional levels and sera levels of IL-17 and IL-23 and IL-9. These are the recent cytokines, IL-17 and IL-9 cytokines. And identification of variants in a COD14 gene in Andhra Pradesh. We have studied this with especially in Andhra Pradesh psoriatic population. So because when you consider the haplotype one, this is very actually is different than different populations, that you know. That's why you have to, while studying about disease, you have to consider that genome constitutions and haplotype of you have to consider haplotype present in the or HLA present in the host or a human. There is association with the genetic, as I said earlier. And psoriasis is immune mediated skin disease. Dermal, mostly you will see the dermal angiogenesis leading to an increasingly complex underlying vascular system, giving the plaques the deep red coloration. Increased vascularity allows for a greater influx of inflammatory cells into the skin, further driving the inflammation. So T cells and a myriad of cells from the adaptive and innate arms of the immune system are present early in the lesions, forming characteristic nests of activated leukocytes in the reticular dermis. Here, there is no role for antibodies actually, psoriasis. That's why we have concentrated only on the cells, especially Th17 cells. T cell infiltrate in the papillary dermis and exclusively CD8 T cell population. The epidermis as such, psoriasis is now generally regarded as a T cell mediated immune disease with a mix of Th1 and Th17 cytokine. So the populations of subpopulations of T cells are differentiated only by means of their cytokine secretion. Okay, you have Th1 cells, Th2 cells, and Th17 cells. There is also another new subpopulation, that is Th9. Okay, because which are alone secretes IL-9. And the TS17 secreting IL-17, whereas TS1 subpopulation of T lymphocyte secretes IL-2, IL-4, and IL-5 interferon gamma. But whereas TH2 able to secrete IL-4 and IL-8. So these are the cytokine and interferon gamma. These are the cytokine. Depending on the cytokine secretion, you can differentiate T cell subpopulations. TS17 cell secretes IL-17 and mostly, and this also having effect on damage effect on fibroblasts and endothelial cells. And we actually worked on keratinocytes because there is a lot of damage on keratinocytes. So keratinocytes isolation and culture was done in our lab. And what the IL-17 actually the bringing about synergistic effects with other cytokines like IL-22 and TNF-alpha, and the chemokines upregulates the chemokines and downregulates some of the chemokines upregulation. There is a, you can see in the slide, but mostly we are focusing on the pro-inflammatory cytokines which are involved in the psoriasis. So those are nothing but pro-inflammatory cytokines and IL-6, TNF-alpha, and IL-17. And these are the cytokines are very, very important. IL-23 environment is very much required for TS17 cells to secrete more and more concentrations of IL-17. And as you know, psoriasis is a disease, immunoinflammatory disease. It's a skin disease, shares immunologic and genetic features with other human autoimmune inflammatory conditions, such as inflammatory bowel disease, rheumatoid arthritis, and multiple sclerosis. Okay, the same mechanism will be followed in all these autoimmune diseases and autoimmune inflammatory diseases. Novel CD4 T cells, TH cells called TS17 cells are very important in the pathogenesis of psoriasis. That's why we have selected TS17 for a study, and TS17 cytokines, most IL-17, IL-23, and the recent cytokine IL-9. Okay, if you go through the literature, you will find very few literature about the IL-9 cytokine. Okay, we worked this project in the year 2016. Okay, isolation of what we have done is isolation of PBMC and keratinocytes from the isolation of PBMC from the blood and isolation of keratinocyte from the skin biopsies. So actually, the GSL College, Medical College, RATHAIAH, and Dr. Surya Hospital, we brought skin samples, skin biopsies. We have ethical clearance for the project, and so that's why we work with skin biopsies and also blood samples. We have isolated the PBMC. There is a protocol using histopath method, and the skin tissues. We actually followed this Nature publishing protocol for the isolation of keratinocytes and culture of the keratinocytes. This is the protocol we followed for skin biopsies for isolating, especially for the keratinocytes. Place skin tissues and poured Hank's balance salt solution containing antibiotics, antimycotics, and kept at 4 degrees centigrade. Place the tissue in uncoated 100 mm bacterial petri dish. Keep moist with a smooth medium, and remove subcutaneous loose connective tissue, easels, and scalpel. Open up or flatten. It is very difficult. Actually, we took training from the dermatologist in the dermis and the keratinocytes. So, and open up or flatten the skin and place the epidermis side down, and use edge of the scalpel and scalpel away tissue and scrap away tissue until only the thin epidermis and dense dermis remain, and place the pieces with the dermal side down in a 60 mm dish containing 5 to 6 ml PBS and with antibiotics and dispace. Dispace is the ingredient which actually overnight we have to keep it for overnight for maceration, proper maceration, because this is the protein which is the tissue, and so that softening of the tissue will happen. And this is kept for 12 to 16 hours overnight. Take overnight digested tissue samples and pieces and separate epidermis and transfer to another dish with the epi-life medium and then place the minced tissue pieces in the falcon tube containing triple E select and incubated 37 days for 45 minutes. Add 20 ml of DMEM and a pipette up and down for the wash the solution through 70 micrometer mesh. Centrifuge 200 grams for 5 minutes and this remove is suspended in the pipe. This is the protocol we followed. Actually, this is the very good protocol. Actually, we got keratinized, wonderful keratinized. So these are the biopsies. Blood samples for PBMC isolation and keratinocyte. These are the cell after culture of keratinocytes and PBMC. We got the supernatant, cell culture supernatant after culturing, and which are stored in -4 degrees centigrade. And then we estimated the levels using the CBA method, cytometric bead assay method using flow cytometry. And IL-17, IL-23, IL levels were elevated subsequently in mild, moderate, severe cases. And these are the results. Actually, they're elevated in the psoriatic patients, both mild and moderate. So mild condition, actually, we cannot see the cytokines, some of the cytokines like IL-6 and so in mild conditions also they're elevated in samples. The individual levels were also found in both keratinocytes and PBMC cultures. And IL-9 associated TL7 response both in blood and tissue. So this is the information. Actually, STAT3 signaling pathway may be important in the clinical manifestations of psoriasis. IL-23, 22 may alter STAT3 progression and affect the skin directly. IL-23 is required, environment is required for the proliferation of the TL17 cells in order to produce more and more IL-17, which is an inflammatory cytokine. Okay, and that is responsible for the pathogenesis of the inflammatory diseases. IL-17 plays a central role for inflammation. And blockade of IL-23 improves psoriasis in big way. Need to find a central pathway for IL-17 also. And come to the other third disease, that is the malaria. I cannot say this is the inflammatory disease. What we have discussed in autoimmune disease, and other disease like the autoinflammatory disease, that is psoriasis. But we could able to found the pro-inflammatory cytokines responses at early stages, pre-erythrocytic stages of malaria. So this project was sponsored by Ph.D. New Delhi, and in the year 2013. And actually, as you know, the malaria pathogenesis is not fully understood with respect to more cytokines, because animal models do not make the main problem. If you want to work with any of the disease, immune-mediated disease, the animal model is very difficult. Obtaining animal model, actually, which do not recapitulate, because the animal models, we don't have exact animal models, are very standard animal, which the animals which is available do not recapitulate exactly human disease. And cytokines play a complete role, have both protective and harmful effects. So parasite antigens released at the cytogamy trigger the release of both pro and anti-inflammatory cytokines. Although the balance between these mediators is critical for parasite control, their role in the pathogenesis of neuronal damage is not yet known. So still we have to find out. So this is the new area I can suggest you if you want to work with inflammatory cytokines in malaria. So this is the the malaria, where you can see this is the pathogenesis. NF-κB signaling pathway. The plasmodium falciparum is the causative agent for the host for the development of malaria. So which binds to the DNA and all like receptor 9. These are the disease damaging patterns molecules which will react with this causative agent, and the signaling happens, and then you will see the synthesis of pro-inflammatory cytokines. This is the pathogenesis of this thing, and we have compared this malaria actually human. We have done with, as I said, animal model as well as with human samples. Using Plasmodium bergae, we induced the Plasmodium bergae strain. We induced the malaria disease, which induces the cerebral malaria in animal model, rat mice. And for the human samples, we have carried out with most of the work, cytokine work. So for that, we have identified the disease samples using microscopy. That is a golden standard model. And we have done with rapid diagnostic test, which is Polyzen kit, which is obtained from the Delhi. And then next, we have done with PCR analysis. This primers was designed, and we have done with so the advantage with PCR analysis is only we need to have micrometers of the sample. That is the thing. So that's why we compared all these three techniques for the identification of the disease pathogen. And this paper is published. So PLH may be a wonderful marker for identification of the biomass and the better parasite biomass and better prognostic indicator than the peripheral parasitemia. And we estimated the PLH levels and using ELISA kit and parasite biomass. And these are the cytokines we have studied. Elevated cytokines, inflammatory cytokines in the plasma of early stages of malaria. What we have identified here is the early stages. The problem with malaria disease is identification of the disease at early stages. So with the help of these markers, so what I thought the identification could be done with the cytokines, but maybe expensive. Using cytokines is not for research purposes. Also, you need to have a funding. But identification is not, but some rapid test has to be identified, designed for the identification of these cytokines, especially pro-inflammatory cytokines identified, elevated at early stages of that means extra erythrocyte stage of early stages of malaria. You have different two stages: erythrocytic and pre-erythrocytic stage. Pre-erythrocytic stage is the early stage, so where we can identify the elevation of these inflammatory cytokines that go. So like that, and cytokine estimation by flow cytometry. And these two cytokines were identified. The low parasite exposure and insufficient sporocytes released to the host, and that is low parasite burden. There are no clinical manifestation. We don't know we have this disease. So without clinical manifestations, there is a usual expression of the cytokines like IL-17, IL-23. So these could be biomarkers for the identification of the disease, so that it cannot progress, it cannot lead to the cerebral. So what we are facing in the Padaru area, they are suffering from mostly cerebral malaria. Most of the diseases during winter season. Malaria is caused by Plasmodium. That is the most prevalent species. Padaru Visakhapatnam. And this we have high parasite exposure. What we have identified at a high parasite exposure, sufficient sporozoites released to the host, and we can also identify the clinical manifestations for the malaria. And manifestation is malaria, chills, and high fever, chills, and all that. And you will see the elevated levels of expression of TCF beta. This is the data actually we found with the disease, human malaria. We also screened some of the extracts for the animal models. Because of lack of time, I cannot on these slides. I'm not able to show all the remaining slides. As you know, this is a very important slide for me, cytokine and immune responses. So always for diseases, whatever it may be, whether it is infection, autoimmune disease, or inflammatory disease, whatever the disease, so you need to understand what is the innate immune response and adaptive immune response. Mostly in innate immune response, that is responsible for whether that decides innate immune response remain within the host, that will decide whether the disease has to progress and disease has to subside. Okay. So this will stimulating natural killer cells. The natural innate immune response is the cells responsible for the development of innate immune response in the host are natural killer cells, dendritic cells, polymorphonuclear cells, and monocytes. And they get stimulated in presence of antigen, like it could be a viral antigen or any other stimulus, extraneous stimulus, and you will get the humoral response, and that is humoral response also developed in the form of increased antibody production. Those are called autoantibodies. That is the response. Not antibody, increased antibodies. Those are called. So that is the antibody dependent. That is, those antibodies are responsible for antibody dependent enhancement. Antibody dependent enhancement of the inflammation. So the site of these cells are also responsible for the formation of cytokines, like most importantly IL-6 plays a very important role for the inflammation, for bringing about the inflammation. In addition, it also produces TNF-alpha, IL-beta, IL-17, and colony stimulating factor, and granulocyte factor is colony stimulating factor, also produced from the Th1 cells and the T cells, and they get depleted, and then you will see the adaptive immune response will be lowered. There is a dysfunction of the adaptive immune response. What is happening in the cytokine storm and septic shock? The adaptive immune response would be declined. Okay. It is not getting up. So instead, the innate immune response is continuing. So that these are the consequences we are facing. Secretion of excess levels of pro-inflammatory cytokines and increased antibody production as a result, increased inflammation and disease progression. Because that is nothing but the cytokine storm. Okay. So there are so many therapeutics we have used. There is no single drug or a single medicine for the treatment of increase of cytokine storm, because the cytokine storm is not because of a single cytokine. It is a multifactorial. You cannot give a single medicine. That's why during second wave we have used so many drugs. We don't know the side effects of most drugs, anti-inflammatory drugs and anti-immunomodulated drugs, whatnot. Subjected for so many other treatments, including corticosteroids. They are anti-inflammatory drugs. So actually, corticosteroids meant for decreasing innate immune response, and whereas FK506, the same type of cytokine storm may arise during the transplantation, graft versus host reaction, and other diseases also. Where you will find immunosuppressive conditions, but where you can use FK506 and cyclosporin A. So for decreasing, you can use corticosteroids, adaptive innate immune response, and for decreasing the adaptive immune response, you can use FK506, and that is the specific immune response, FK5 cyclosporin immunosuppressive drugs. So that's how we can differentiate if you understand the detailed mechanism of immune response. So you have the ability, or you have the capacity to differentiate what drugs are suitable for this disease particularly at that point. That's why they have suggested, the medical people suggested steroids. This is second phase, is the steroid responsive phase. Why they have decided this, they know. So this is the purely innate immune prolongation of the innate immune response. There is no raising of adaptive immune response. If adaptive immune response raises, immediately it controls over, takes over the disease, it takes over the virus, but it is not happening. It is not happening. Adaptive immune response, specifically only continuing of adaptive immune response. That so therapeutics, vaccines or therapeutics was the direction of this only to promote the adaptive response from the T cells. has to be developed. So this is another slide, metabolic engineering. It is a marine organism. This is the trend changes now, completely changed. Instead of screening, it is very difficult. Actually, I face whether you are facing or not, I don't know. But screening of marine plant products is very easy when compared to marine natural products. In fact, the marine natural products, we brought very good samples from the islands because JDSA people help at the Port Blair, they help for collection and all that. Some of the unexplored species also we brought from Andaman Nicobar. So we could able to do some of the, roughly 20 or 18 extracts. So this is a very difficult process. Actually, we are unable to do both the balance immunological experiments and pharmacy, isolation and isolation process and chemistry. So we're able to screen some of the compounds against these diseases. And anyhow, nowadays the trend has been completely changed. You might have heard about metabolic engineering. First, we need to have a marine organism, and what are the compounds, powerful compounds present in that. So you need to understand the omics, genomic data, proteomic. You need to subject it for proteomic, transcriptomic, and metabolic studies. So that if you, after understanding this metabolic pathways, so you will understand what is the gene that is able to secrete this desired product and responsible for the production of the compound. And then you can genome scale metabolic model can be designed, and product optimization can be done, and then the knockout strains, metabolically important genomic knockout strains can be developed in order to increase the production of the compound. Because most of the marine, the problem with the marine organisms and culture cannot be possible with culturing, and some of the organisms we cannot raise because more precious. So for those organisms, you can isolate the gene, desired gene, and can put it into the important strain, and we can develop a model, and then can produce a product optimization can be done. That is the objective of the. And marine compounds that are used as drugs, microorganisms, plants, and animals are synthesized in the small amounts and are very difficult to obtain in large quantities. This is the area where metabolic engineering comes into play. Use of metabolic engineering, we can model organisms using existing metabolic reconstructions to discover gene knockouts, which could improve the yield of the products of interest. So this thing actually I believe because achieve. So first you have to identify the root cause for the treatment. Then only you can treat the patients or disease. First, understanding the disease is very important. For that, you need to have a knowledge on molecular biology, microbiology, immunology, and the other areas also you need to have. Then only you can work on that. Achieve mechanism-based specific treatment for immune inflammatory diseases. The need of our.
Thank you very much, ma'am.
Thank you for the kind attention.
It was a very informative lecture. Now, it's a time for just question and answers because we are running out of time. We would like to allow a few questions. If any of the participants want to ask any questions and interact with ma'am, like you can unmute yourself one by one and you can ask, or you can just paste your questions in the chat box also.
Any questions?
Yeah, if nobody has right now, if they don't have any questions, like you can mail to the ma'am, we will just forward ma'am's ability. Thank you, ma'am.
Ma'am, they are in the chat box.
Thank you.
Purna madam, Can I ask one question, ma'am?
Yeah, sure, ma'am. Sure, ma'am.
Please.
Madam, can we use any antibodies against these cytokines that are produced in allergic conditions or autoimmune disorders?
Okay, you can monoclonal antibodies are developing
Okay.
against these cytokines. So the treatment, as I said, for IL-17, IL-6, and TNF-alpha, for all these monoclonal antibodies have been developed for the treatment. They are specifically acting on them. Nowadays, for COVID also, they are giving against for IL-6.
Okay, thanks. Okay, thank you, ma'am.
Shall I leave? Purna?
Please wait, ma'am. Yes, there is. May I invite any head institutions to make any good remark? Srinivas Babu, sir. Sir, would you like to? Okay. So remaining participants, please mute yourselves. Okay, ma'am. Thank you. Now it's time for vote of thanks, ma'am. Last one. Now may I invite Dr. D. Vasudha, Associate Professor from Department of Pharmaceutical Chemistry, Minister of Pharmacy, to deliver her vote of thanks. Over to Dr. D. Vasudha.
Thank you, ma'am. Good evening, all. As all good things come to an end in life, so is the webinar. On behalf of Vignan Institute of Pharmaceutical Technology, I, D. Vasudha, Associate Professor, take this opportunity to propose vote of thanks. First of all, I would like to thank today's speaker, webinar speaker, Dr. Aparanji Posarla, for enlightening us with knowledge. We are really enlightened with your knowledge and presence, madam. Thank you very much, ma'am, for accepting our invitation in spite of your busy schedule. I extend my deep gratitude to our respected chairman, sir, Dr. L. RATHAIAH and L. KRISHNADEVARAYULU, Vice Chairman, Vignan Group of Institutions. Next, I would like to thank Dr. K. RAMAMURTHY NAIDU, Chancellor, VFSTRU, Dr. M.Y.S. PRASAD, Vice Chancellor, VFSTRU, Dr. M.S. RAGHUNATHAN, Registrar of Vignan University, for providing this opportunity to organize the webinar. Our heartfelt thanks to head of the institutions, Dr. Y. SRINIVASA Garu, Principal, Vignan Institute of Pharmaceutical Technology, Dr. P. Srinivas Babu Garu, Vignan Pharmacy College, Guntur, Dr. M. Srinivasmurthy Garu, Vignan Institute of Pharmaceutical Sciences, Hyderabad, Dr. C.H. Jithendra, Faculty of Pharmacy, VFSTRU. A special thanks to the organizing committee, teaching and non-teaching staff for their support and coordination. Our heartfelt thanks to our students for active participation. With these warm words and a kind message, we move to the end of today's webinar. Thank you, ma'am. And one thing I want to tell from my side, after seeing your resume and everything, most of our pharmacy students, most of them are all girls, ma'am. So we really inspired with you.
Thank
with
you.
Your presentation and everything.
But you didn't pose any question.
No, ma'am, questions are
Whether
there
it
in the
is
chat box.
understood or not relevant.
Ma'am, questions are there in the chat box, ma'am.
Okay, chat box. Okay.
Yeah, questions
Lack
are there
of
in the
time.
chat.
Okay.
Yeah, lack of time.
Okay,
Questions
anyhow,
are there in
thank
the chat.
you. Anyhow,
Now, most of our students, they get inspired with your presentation, especially by seeing your resume and everything, like the awards you got. Really, we are enlightened with your knowledge, ma'am. Thank you very much, ma'am, for accepting our invitation.
Thank you for giving me this opportunity. Purna, thank you.
Thank you, ma'am. Thank you so much.
Thank you, ma'am.
Yes, ma'am. We can leave, ma'am. Thank you, Vasudha. Thank you very much, ma'am. Ma'am, due to the time constraint only, like most of the questions were like,
okay, even
we
I
will mail.
see that.
Yeah. Thank you, everyone. Thank you, one and all. Thank you so much, ma'am.
Okay.
You can leave, ma'am. We are ending now.